And so this is part of an ongoing collaboration with Andy Berglund's lab at the RNA Institute at the University at Albany. So as we know, pathogenesis in myotonic dystrophy stems from the production of toxic CUG and ccUG expansion RNAs. And so these toxic RNA molecules, once they're formed in the cell, gain a number of toxic downstream consequences. And so some of these are outlined in this schematic. And so in terms of thinking about therapeutics, these toxic processes could be targeted individually. However, approaches that target the production or accelerate the degradation of these toxic RNA molecules should theoretically circumvent all these downstream toxic consequences to the cell. And so with this in mind, when I was a postdoctoral trainee in Andy Berglund's lab, and also working in collaboration with Eric Wang's lab at the University of Florida, and particularly Ona McConnell in Eric Wang's lab, we set out to develop a screening system or a platform where we could selectively monitor expression of the toxic CUG RNA molecules relative to a control, such that we could screen, for example, for small molecules and biological agents that would selectively reduce the toxic RNA molecules, which are our target. And so towards this end, what we did is we developed a screening platform, and we opted for HeLa cells just for the ease of culture and scalability and cost for larger screens. And then we utilized constructs that were previously developed in Tom Cooper's lab that contain a 480 interrupted CTG repeat tract within exons 11 to 15 of DMPK. So there's some native genetic context for the repeats. And we paired these with control templates that lacked that repeat, but otherwise were identical. And so we also added some unique sequences to each of these templates so that we could distinguish the RNA that's generated from the toxic template versus the control template. And so this would give us a means to measure for selective effects on the toxic RNA. And so these constructs, these paired constructs, were integrated into the HeLa genome. And this occurred randomly using the piggyback transposon system. And a number of clonal cell lines were identified that robustly express both the toxic RNA as well as our control RNA at roughly comparable levels. And so we identified a number of clones that showed expression of both. And again, we can probe for the presence of the toxic RNA and the control RNA separately or together. And importantly, these cells recapitulate some of the molecular and cellular hallmarks of myotonic dystrophy that we know. For example, the formation of CUG ribonuclear foci, as well as missplicing of MBNL-mediated pre-mRNAs. And so we've previously applied these cells in small molecule screens where the cells are cultured in multi-well format and treated with small molecule libraries, following which multiplex RT-qPCR is performed in order to identify abundance of the toxic RNA relative to the control. And so we've published some of this work. And so from this work and small molecule screening, we've identified compounds that selectively reduce these toxic CUG RNAs in various DM1 cell lines, as well as HSALR mouse model. For example, we published that unexpectedly microtubule inhibitors were a class of compounds that could selectively reduce the CUG RNA expansions, RNA levels. And more recently, Subodh Mishra, who's a research scientist in Andy Berglund's lab, has applied the screening platform to natural compound libraries to identify compounds with favorable toxicity profiles. We know that microtubule inhibitors have toxicity issues. So in more recent screens, he's been he has identified some promising natural compounds. And so he'll be telling us more about that tomorrow in his poster session, as well as his flash talk. And so while these small molecule screening projects are ongoing, it also occurred to us that we could apply these screens to we can perform genetic screens using these cell lines. And so this could be accomplished using siRNA libraries as well as CRISPR-Cas9 guide RNA libraries. And so here, in the same way, you can treat your HeLa DM1 cells and then identify genetic factors that, when modulated, give rise to modulating toxic RNA levels. And so today I'll tell you about an unpublished study where we carried out our genome scale siRNA knockdown screen using the HeLa DM1 cell line. And so we opted for a knockdown to start with over a knockout screen because we were interested in both essential as well as non-essential genes whose expression levels may modulate the toxic RNA. And so just to walk you through the screen that we did, we used the HeLa DM1 cell line where they were reverse transfected with an siRNA library. And so this is a commercially available siRNA from Sigma Aldrich. The E just means that it's an enzymatically derived siRNA library. And so these cells were treated in 384 well format. And so the library has 44 384 well plates that encompass just under 17,000 genomic targets. And so following about 72 hours of culture, where each well contains a single siRNA, multiplex RT-qPCR is performed, and then we measure the effects on the toxic CUG RNA levels relative to the control. And as a positive control throughout the screen, we used an siCUG to ensure that we were getting knocked down throughout the screen. And so following completion of the screen, we can see that we have a robust approximately 50% reduction of CUG levels. And again, this is relative to the CUG zero control. And so just to summarize what we identified from this screen is we filtered for hits based on the siCUG positive control of 0.5. So all hits that reduced relative CUG 480 levels below 0.5. And we identified 380 hits from the screen at roughly 2% hit rate. And we also filtered for hits that elevated or stabilized CUG 480 levels. And so we filtered with a cutoff that gave us approximately an equal number of hits as the reduction. And however, with the elevated levels, we didn't really have a positive control. So we just filtered to identify hits based on that cutoff. And so what was interesting is when we plugged all our hits into, for example, gene ontology analysis from PantherDB, the hits were all generally enriched for splicing for the most, or for the top 10 at least, were all splicing related terms. And interestingly, the top 5 were all spliceosome terms. However, to probe a broader set of hits, we looked at the nucleic acid metabolic process. And so this was a broader category, which was at the 10th position. And it encompassed most of the preceding hits, but it also had a more diverse set of hits, including, for example, DNA repair proteins, as well as RNA modification enzymes, and so on. And so we performed a secondary screen with the nucleic acid metabolic process category, which contained about 100 genes. And we carried this screen out in 2 independent HeLa DM1 clonal lines, just to ensure that the random integration of the constructs didn't influence the hits that we picked up. And so, first of all, it was interesting that actually none of the hits that we initially identified that elevated the CUG 480 levels were recapitulated in both cell lines. So not sure why. But when we looked at hits that reduced the CUG 480 levels, we picked up about 20 hits that were robustly reducing the relative CUG 480 levels. And this is with an independent set of siRNAs. So we noticed that not all of them hit our initial threshold of 0.5. But what was interesting is out of the top 10 hits, 8 of them were all core components of the spliceosome. So this was really interesting for us and unexpected. And out of these top 20, we also picked up hits that were previously identified that were related to DM, for example, DDX17 and HNRNPA1. But the top 8 were all core components of the spliceosome. And so it was interesting that knocking down these core components of the spliceosome actually led to a selective reduction in CUG 480 levels. So we wondered if there was any correlation with expression of any of these factors in DM patients and the expression of DMPK, which carries the expansion. So we analyzed the expression of these top 8 spliceosomal hits in DM patients on dmseq.org. So this is TA RNA-seq data. And what we noticed that there was a significant positive correlation between our top 2 hits, SNRPD2 and SF3B5, and DMPK expression. And furthermore, we also saw that expression was significantly lower in DM1 patients compared to controls. And so while this is correlative, it was interesting that there was this correlation and could plausibly indicate that there may be some compensatory expression changes that could potentially mitigate the CUG expansion levels by modulating DMPK. But again, correlative, so you have to take that with a grain of salt. And so it was interesting that we identified spliceosomal components. And so as it turns out, splicing is actually very complex. There's a lot of moving parts, and the spliceosome itself is composed of various subcomplexes. And so in a splicing reaction or a splicing cycle, multiple components of the spliceosome have to assemble and interact, get remodeled, and so on in order to excise the intron and ligate the exons. And furthermore, we know that alternative splicing regulators like MBNL1 have to coordinate the interaction with the spliceosome by binding to the appropriate pre-mRNA sites to either promote inclusion or exclusion of exons by inhibiting or enhancing spliceosome interaction with the pre-mRNA sites. And so we know that this occurs while MBNL levels fluctuate over development. And furthermore, we also know that MBNL1 is sequestered by toxic CUG RNA in DM patients. So this equilibrium is disrupted in DM. And so with that in mind, we wondered, because we initially picked up only a subset of the spliceosome that reduced these relative CUG 480 levels. So we then carried out a more targeted screen where we selected a compact panel of 12 factors that represented the various subcomplexes of the spliceosome, and then performed the same analysis looking at the effect on CUG 480 levels, but also MBNL-mediated alternative splicing. And so what was interesting is, in general, we did see that many of these spliceosomal components did selectively reduce the CUG 480 levels, but this wasn't universally true. So there were some that did result in the selective reduction and others that didn't. And then when we looked at MBNL-regulated splicing, we also saw again that some of these components did rescue splicing. So if we're looking at this MBNL1 exon 5 event, normally muscleblind excludes this exon. And so you have lower levels in non-DM cells. And in the HeLa DM1 model, because the MBNL1 is sequestered, you now have more inclusion because it can't regulate. But then when you knock down these spliceosomal components, you can see that actually some of these events are rescued. But again, not all the components did show this rescue. And this was also true of both inclusion and exclusion. So here I'm showing NUMA1 exon 2. Normally, MBNL1 here promotes inclusion. And in the DM cells, you see that it is included less frequently due to the sequestration. And then when you knock down your select spliceosomal components, we see that again, there's rescue. But again, this was only occurring with a subset. And interestingly, when we looked at the effect on CUG 480 levels relative to MBNL1 splicing, the expectation being that depleting these levels should result in a rescue of missplicing, we saw that this wasn't actually strictly the case. So if you look at, for example, SNRP-A, which is part of the U1 SNRP, it didn't have a significant effect on the relative CUG 480 levels, but it did significantly rescue missplicing. So it appeared that these two events, though related, weren't dependent. And so we wanted to investigate the effects of modulating spliceosomal components in greater detail. So we selected one of our top hits, SNRPD2, and modulated its expression in DM1 and control patient fibroblasts. And so first we confirmed that we were leading to a basically modest reduction in SNRPD2 levels. And so what we saw is with about 40% reduction, it was sufficient to significantly reduce DMPK expression, albeit it was more modest in the fibroblasts. And interestingly, in the control cells, we did see that it was being affected, although it wasn't significant in this case. And we were also significantly rescuing missplicing of both inclusion and exclusion in DM1 patient fibroblasts. But interestingly, again, we saw that there was an effect in the control fibroblasts, suggesting that consistent with what we saw in our HeLa DM1 cell model, that this wasn't dependent on the expanded cUGRNA. And just to confirm that we weren't observing these effects just as a consequence of general splicing failure, we treated both control and DM1 cells with isokinkatine, which is a pre-mRNA splicing inhibitor and a generalized inhibitor. And so we confirmed splicing failure across this treatment dose range using this DNA JB1 intron 2 retention event that was previously reported. And then we looked at both DMPK expression and MBNL mediated alternative splicing. And we didn't see the same effect on either DMPK expression in either the control or the DM1 fibroblasts, nor did we see any effect on insulin receptor splicing, for example. And in fact, if anything, it looked like it was actually being exacerbated in the dose range. And so it did appear that the effects that we see from modulating SNRPD2 levels are independent of just inducing splicing failure, because we are knocking down a core component of the spliceosome. And to more broadly interrogate the transcriptomic effects, we then performed RNA sequencing following knockdown of SNRPD2. And then we assessed the major categories of alternative splicing. And so what we noticed is when we knocked down SNRPD2 in both control and DM1 fibroblasts, we saw that it largely impacted skipped exon alternative splicing events. So this is consistent with previous studies showing that actually modulating core spliceosomal components affects alternative splicing independent of splicing failure. So a number of years ago, this was quite surprising because the spliceosome was always seen as important for basal splicing, and it wasn't really a regulatory entity. And so when we looked at the individual events and we measured the absolute delta PSI for these categories, we noticed that across both the control and DM1 cells, you generally had a comparable delta PSI change when we knocked down SNRPD2. So this didn't indicate a general difference between the DM1 and control cells. However, when we did look at MBNL regulated alternative splicing, we saw that the delta PSI changes were greater in the DM1 cells. And here, consistent with MBNL being sequestered by the toxic RNA and that regulatory window being more sensitive in DM. And so because muscle symptoms are a central feature of myotonic dystrophy, we also wanted to confirm these results in muscle cells. So we performed SNRPD2 knockdown in myoblasts. And here we actually noticed that the effects were even more dramatic, where even just about a 25% reduction in protein levels significantly reduced DMPK expression by about 50%. And interestingly, this occurred in both control as well as DM1 myoblasts. So unlike in the HeLa DM1 cell model where it was showing a selective effect, here it was showing general DMPK reduction. And again, we confirmed that missplicing of MBNL regulated events were significantly rescued in control in the DM1 myoblasts, and there didn't seem to be a dramatic effect in the control cells. And so when we looked at our RNA-seq expression data, I didn't go into it in the interest of time, but we did notice that MBNL1 expression was elevated in the fibroblasts. So we looked at RNA expression for MBNL1 and saw that it was significantly elevated in the myoblasts. However, when we looked at the protein levels, it didn't appear to be boosted as a result of SNRPD2 knockdown. So the effects we're seeing were not due to, at least not simply due to MBNL1 protein levels being elevated as a result of SNRPD2 knockdown. So that's currently where we're at with the study. And based on our data, our working model is basically that normally MBNL1 is in equilibrium with the spliceosome and has to be in order to regulate alternative splicing of a number of events. And in myotonic dystrophy, because of the toxic CUG expansion RNA, MBNL becomes titrated away from its normal alternative splicing role. So this system is in imbalance. So now what we find with our work is if you modulate the spliceosome, you may actually be able to partially restore some of that imbalance. And so while we think a lot about this side of the scale, I think it may be relevant to think about this side of the scale as well. And so from this work, there's a number of unanswered questions that we're actively pursuing. For example, we don't actually know what the mechanism impacting DMPK expression is. So this is something that we're working on. And so what's interesting is when we look in the HeLa DM1 model and compare that to the myoblasts or fibroblasts, what's distinct is in the HeLa DM1 cell model, the control is a 0 repeat template. Whereas in the fibroblasts and myoblasts, there is a repeat. It's not expanded, but it's there. So we're wondering actually if the repeat may play some cis regulatory role in expression of DMPK. So this is something that we're pursuing. Another question is, what happens to MBNL1 binding when we modulate spliceosome components? Because in this study, again, we are knocking down SNRPD2, for example, and other spliceosome components. But what we haven't done is overexpressed. So the prediction would be that you could potentially tip the scale in the other way and actually exacerbate some of the roles. And also, it's not clear why only certain spliceosomal components show this regulatory window. We tested other ones. There may be others, but not all of them seem to. And finally, we're interested in whether there are any variants in SNRPD2 or other spliceosomal components that may potentially correlate with DMPK expression and or MBNL-mediated spliceopathy. So I'll end there and just move to my acknowledgements. I just want to acknowledge there were 3 main players who drove a lot of this research. So Gis Maria Lewis is a postdoctoral fellow that's co-advised by Andy and myself. And then Jesus Frias is a PhD student in Andy's lab, and then Jacob Schroeder is a PhD student in my lab. And I'd also like to acknowledge Claudia Lennon and Emily Davey, who did the dmseq.org analysis, and all members of the Reddy and Berglund labs, particularly to Andy for his support and guidance, and then our collaborators and funding sources. And also a particular thanks to Myotonic Dystrophy Foundation for supporting a lot of our work. A number of us from our labs have been MDF fellows, so we're greatly appreciating of that. So thank you, and I'll take your questions.
See a question back here, and then we'll get you comment.
Very interesting work. I was wondering if there was any interest or thought to try and expand your screen to involve genes that wouldn't be expressed, say, in like a HeLa cell line that might be tissue specific, such as doing like a CRISPR activation screen in the same way with your lines.
Yeah, so that is something that we're definitely interested in. Different cell lines, different types of screens, activation screens as well. Initially, we were looking at sort of one of the challenges is in our system, we're reliant on arrayed screens from our readout. So that just makes it very laborious, expensive. And so, yes, we definitely do want to expand into other systems.
That's beautiful work. That's really exciting. And I thought it was that it looked like DMPK, the expanded DMPK pre-mRNA is not going to be processed well, and that would explain the results in your screen. And then I was excited about you could use splicing inhibitors, and that was going to be great. And you did all that. And then in the muscle cells, both the control cells and the expanded cells shows the same thing. So it still is unclear what's the problem with the expanded DMPK. Why does that go down in your screen? I guess it still could be that in that screen, because of levels of expression or something, that the pre-mRNA is maybe overwhelming the system, and it may be more sensitive. I don't know.
Yeah, I mean, those are good thoughts. So we are sort of thinking about ways to probe that mechanism. I think the one thing that stands out between the two systems is with the HeLa line where we're comparing to a 0 repeat control. So I wonder if there's something to having some repeats, whether they're expanded or not. And so that's something that we'd like to test. So we're also interested, for example, if we look in sort of the knock-in mouse model that Maury Swanson's lab developed, where the mice have don't have the repeat tract versus having a knock-in repeat, whether we see similar effects. But there are certainly other mechanisms that could be at play. So we're thinking about ways to address that.
I still wonder if it wouldn't be worth trying a couple more splicing inhibitors that might hit other targets.
Yeah, absolutely. I think that's a good idea. And the other thing is when we actually recently just finished doing foci analysis on the myoblasts, and we know that the foci do go down. So it's not selective for DMPK, but we know that we're at least hitting the expanded repeat as well. But yeah, trying other splicing inhibitors would be good. Different, because I think they're going to be different targets in the splicing cycle. So.
All right, let's thank Kaalak one more time.